from news-medical.net. What are coenzymes? Coenzymes are essential components of the many metabolic processes that sustain life on the cellular level. What are coenzymes? A coenzyme is defined as an organic molecule that binds to the active sites of certain enzymes to assist in the catalysis of a reaction. More specifically, coenzymes can function as intermediate carriers of electrons during these reactions, or be transferred between enzymes as functional groups. For example, during the conversion of pyruvate to acetyl coenzyme A, or CoA, several coenzymes, including free CoA, thiamine pyrophosphate, TPP, lipoic acid, flavin, adenine, dinucleotide, two cellular redox enzymes, including oxidized nicotinamide, adenine, dinucleotide, NAD, and reduced nicotinamide adenine dinucleotide, NADH, are required. Important coenzymes. Coenzymes, which are often vitamins or derivatives of vitamins, therefore play a crucial role in the regulation of most enzyme activities. In addition to some of the aforementioned coenzymes that are involved in the generation of the energy molecule, adenosine triphosphate, ATP, several other coenzymes are considered to be fundamental to the existence of all living cells. These include two additional redox coenzymes of oxidized nicotinamide adenine dinucleotide phosphate, NADP+, and its reduced counterpart of NADPH, as well as other energy coenzymes like adenosine diphosphate, ADP, and adenosine monophosphate, AMP. Several coenzymes also function as antioxidants to eliminate reactive oxygen species, ROS, some of which include oxidized glutathione, GSSG, and reduced glutathione, GSH. Coenzymes and citric acid cycle. Within the body, glucose is required for the synthesis of ATP, which functions to store and transfer energy to cells throughout the body. Glucose can be metabolized through an anaerobic process known as glycolysis, or an aerobic process known as the citric acid cycle. Although glycolysis does not require the input of oxygen to produce ATP, this reaction is limited in its ability to harvest a significant amount of ATP that is available from glucose. Comparatively, the citric acid cycle, which requires the input of oxygen, can produce more ATP molecules than glycolysis and therefore supply more energy to support many metabolic processes required to sustain life. In fact, the citric acid cycle, in conjunction with oxidative phosphorylation, is responsible for producing more than 95% of the energy used by aerobic cells in human beings. As previously mentioned, the citric acid cycle, which is also referred to as the Krebs cycle or the tricarboxylic acid cycle, TCA, is central to all metabolic processes that occur within the cell. TCA begins with the condensation of the coenzyme acetyl-CoA to citrate. Citrate then undergoes dehydration to produce cis-aconitate, which is then rehydrated to form isocitrate. Catalyzed by the enzyme isocitrate dehydrogenase, isocitrate is converted through a two-step reaction to aketoglutarate. These irreversible reactions result in the formation of NADH and carbon dioxide, CO2. Once a ketoglutarate is formed, it then undergoes an oxidation reduction reaction to form a 4-carbon compound known as succinyl-CoA, while simultaneously reducing NAD plus into NADH. Succinyl-CoA then undergoes an energy-conserving reaction to form succinate, during which guanosine diphosphate, GDP, is phosphorylated to guanosine triphosphate, GTP. Once GTP is formed, it readily transfers its terminal phosphate group to ADP in order to form a new ATP molecule. After succinate has been formed, the enzyme succinate dehydrogenase is used to remove 2 hydrogen molecules from succinate to form a new molecule known as fumarate. The formation of fumarate allows for FAD to accept the hydrogen molecules, thus forming FADH2. From here, FADH2 can enter the electron transport chain, which results in the formation of 2 new ATP molecules. Returning back to the citric acid cycle, fumarate is hydrated to form L-malate, which then undergoes a dehydrogenation to form oxaloacetate. The oxidation reduction reaction that is responsible for the formation of oxaloacetate also reduces NAD plus to NADH. Taken together, a single citric acid cycle produces 3 NADH molecules, 1 FADH2 molecule, 1 ATP molecule, and 2 CO2 molecules, since a single glucose molecule will form 2 pyruvate molecules, both of which will undergo their own metabolism by the TCA. The production of these high-energy products is doubled. Furthermore, the energy-rich molecules produced by TCA are crucial to the downstream production of ATP via the electron transport chain. Coenzymes and diseases. In addition to the TCA, other metabolic processes involved in programmed cell death, calcium homeostasis, the production of reactive oxygen species, ROS, and oxidative stress all occur within the mitochondria. The dysfunction of coenzymes, as well as any other mitochondrial components, can directly alter a wide range of both anabolic and catabolic pathways that can contribute to the development of various disease states. Several different neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, and Huntington's disease, are associated with altered mitochondrial dynamics. Similarly, alterations in the redox potential of the mitochondria are also involved in numerous cardiovascular diseases, including cardiac hypertrophy, myocardial ischemia, reperfusion injury, and hypertension. To date, there is a limited amount of data available to correlate the direct role that coenzymes have on these diseases. However, their importance in the regulation of enzymes that are known to contribute to certain disease states allow researchers to conclude the inevitable impact that coenzymes also have on these health conditions. While this lack of understanding remains, several clinical studies have already found that incorporating certain coenzyme therapies can improve the occurrence of positive outcomes in the treatment of certain cancers and other diseases. As more research is conducted to further advance the understanding of what molecular roles coenzymes have in certain disease states, researchers are hopeful that these efforts could result in the identification of novel therapeutic targets. Last updated February 17, 2021.